Hello students, we are going to study hemostasis and thrombosis. So in normal hemostasis, the important is integrity of small blood vessels. There should be adequate number of platelets, normal amount of coagulation factors and coagulation inhibitors, along with adequate amount of calcium ions in the blood. In normal hemostasis, following injury, there is transient arterial vasoconstriction, which is mediated by neurogenic mechanisms, and endothelin, which is secreted by the endothelium and is a potent vasoconstrictor. So, first step in hemostasis is vasoconstriction, which is transient, mediated by neurogenic mechanism and endothelin released by the endothelial cells, which will cause vasoconstriction at the site of injury. After transient vasoconstriction, primary hemostasis will occur. The endothelial injury, which exposes the highly thrombogenic extracellular matrix, that will facilitate the platelet adhesion and platelet activation. Platelet activation include the change in shape and release of the secretory granules, leading to formation of primary hemostatic plug. So following injury, first there is vasoconstriction. After that, the endothelial injury, it will expose the collagen extracellular matrix that will facilitate the platelet adhesion and platelet activation in the form of shape change and release of secretory granules. Then recruitment and aggregation, forming the hemostatic plug. In circulation, platelets are membrane-bound discs expressing a number of glycoprotein receptors of integrin family. They contain two types of granules, alpha granules and dense granules. After formation of primary hemostatic plug, there is secondary hemostasis. The tissue factor, that is factor 3, is also exposed at the site of the injury. It is a procoagulant secreted by the endothelial cells. It will lead to thrombin generation that will cause fibrin deposition. The fibrin meshwork and additional platelet reactivation that will lead to secondary hemostasis. So in secondary hemostasis, tissue factor is important factor which will lead to thrombin activation leading to fibrin deposition. The role of endothelial cells in hemostasis. Endothelial cells are key player in regulation of hemostasis. Thrombus formation is determined by the balance between antithrombotic and prothrombotic activities of endothelium. Endothelial cells, they are antithrombotic. Endothelial cells prevent thrombosis by production of factors that block the platelet adhesion and aggregation, inhibit the coagulation, and lyse the clots. So it has antiplatelet effects, anticoagulant effects, and fibrinolytic effects. Coming to antiplatelet effects of endothelium, intact endothelium prevent platelet engaging with ECM. Non-activated platelet do not adhere. If platelets are activated, then prostacyclin and nitrous oxide produced by the endothelial cells impede platelet adhesions. Endothelial cell also produces ADPase, which degrades the ADP, so it inhibits the platelet aggregation. Anticoagulant effects, they are mediated by the endothelial membrane by producing the heparin-like molecules and thrombomodulin along with tissue factor pathway inhibitor. Fibrinolytic effects. Endothelial cells, they will synthesize the tissue type plasminogen activator that cleaves the plasminogen into plasmin. Then plasmin cleaves the fibrin to degrade the thrombi. Prothrombotic properties of endothelial cells. Trauma and inflammation of endothelial cell, it induces a prothrombotic state. Proplatelet adhesion is von Willebrand factor. Procoagulant activated to secrete tissue factor, and antifibrinolytic effect is inhibitors of plasminogen activators that depress the fibrinolysis and it favors the thrombosis. Now thrombosis. What is thrombosis? Thrombosis is a pathological process which denotes the formation of clotted mass of blood within the uninterrupted vascular system. So important is pathological process, formation of clotted mass of blood in the uninterrupted vascular system. It is also considered an inappropriate activation of normal hemostasis process. Three primary abnormalities that will lead to thrombus formations, also known as Virchow's triad. These are endothelial injury, stresses or turbulent blood flow, hypercoagulability of blood. So, Virchow's triad, thrombus formation due to abnormal blood flow, endothelial injury, and hypercoagulability. First factor, endothelial injury, is important for thrombus formation in heart or the arterial circulation. Example, cardiac chambers in myocardial infarction, over ulcerated plaques in atherosclerotic arteries or in vasculitis. Another factor is hypercoagulability. It is less frequent contributor to thrombotic states. Any alteration of the coagulation pathway that predisposes to thrombosis, like primary cause or genetic, when there is factor V gene mutation and prothrombin gene mutations. Secondary cause is acquired in case of immobilization, obesity, cancer, atrial fibrillation, MI, tissue damage after surgery or burns. Third factor of Virchow's triad causing thrombosis is alteration in normal blood flow. When there is turbulence in blood flow, either due to endothelial injury, local areas of stasis, or disrupt lamellar flow, that will lead to movement of platelets from center of the flow to the vessel wall, prevent dilution of the activated clotting factors by flowing blood, will cause slow down the inflow of clotting factor inhibitors, and it promotes endothelial cell activation. So, morphology of thrombi. Thrombi may form anywhere in the cardiovascular system within the cardiac chambers, arteries, veins, or capillaries. They are of variable size and shape. When thrombi are formed within cardiac chamber or aorta, they may have apparent laminations called the lines of Zahan. So, there are differences between arterial thrombi and venous thrombi. The arterial thrombi, they are formed in rapidly flowing blood of arteries and heart. And venous thrombi, they are formed in slow moving blood in veins. Arterial thrombi are commonly seen in aorta and coronary arteries, while it is seen in superficial varicose veins, deep leg veins, popliteal veins, femoral vein, iliac veins. Arterial thrombi is formed following the endothelial injury, while venous thrombi is due to venous stasis. Arterial thrombi, they are usually mural, not occluding the lumen completely, while the venous thrombi are occlusive. Arterial thrombi are gray-white and they are friable and made up of tangled strands and layers of fibrin, platelets, and mashed in clot. While the venous thrombi, they are red color red-blue in color as formed of richer admixture of red cells. Arterial thrombi, they will cause ischemia, while venous thrombi, they will lead to thromboembolism. So this picture is depicting the types of thrombus. Here it is occlusive thrombus, occluding the whole lumen. Then the embolus, detached from the thrombus. Then mural thrombi, which is not occluding the lumen. Now the important part of thrombus, fate of thrombus. So thrombus may propagate. So first is propagation. A thrombus may enlarge in size due to more and more deposition from the constituents of flowing blood and eventually cause obstruction of some critical vessels. It may embolize. So embolization. Fibril thrombi may get detached from the vessel wall and may dislodge the distal sites in the vascular tree. Third is dissolution. They may be removed by fibrinolytic activity. Fourth is organization and recanalization. If the thrombus is not removed, it starts getting organized. Phagocytic cells come and phagocytose the fibrin and cell debris. Proteolytic enzymes liberated by the leukocytes, endothelial cells, they start digesting coagulum. The capillaries grow into thrombus and re-establish the blood flow called recanalization. So fate of thrombus is either it may propagate or it will resolve or it will cause thromboembolism, detachment of thrombi, then organization and recanalization, formation of capillaries. Now coming to differences between antemortem thrombi and postmortem clots. The antemortem thrombi, they are dry, granular and firm, while postmortem clots, they are gelatinous, soft and rubbery. Antemortem thrombi, they are adherent to vessel wall, while they are weakly attached to the vessel wall. Antemortem thrombi surfaces contain lines of Zahan, while postmortem clot, the surface is chicken fat yellow, covering the underlying red currant jelly. Antemortem thrombi may or may not take their vascular contours, while postmortem clots, they take the shape of the vessel or its bifurcation. So pale thrombus, it is formed by platelets continuously activated and fibrin precipitation. It results from severe injury or continued blood flow disturbance. So it is comprised of platelets and red blood cells. While the red thrombus, fibrin and red blood cells predominate, more common in vein. In pale thrombus, fibrin deposition is prominent, while in red thrombus, fibrin and red blood cells predominate. Line of Zahan, there is high rate of fibrin formation. Pale thrombus traps the RBCs followed by another layer of platelets bound by fibrin. Thank you.